Большое спасибо. Спасибо. Guys, start coming. Start collecting for a lecture, guys. Guys. Oh, hello. First students are coming up already to listen to a lecture. Good morning, guys. Today we are going to discuss some unconventional bacteria. What does it mean, unconventional bacteria? They don't look like normal bacteria that we studied up to here. So we studied bacteria that are free living bacteria. I mean, they are parasitic, but they don't need the human cell to develop themselves. So they are traditional bacteria that may grow on the surface of the solid medium or in the liquid medium in the laboratory. So and can multiply the air by binary fission and can develop energy by themselves and proteins and everything. So we have finished lectures for traditional standard bacteria. So and next two lectures will be devoted to bacteria that are not normal in brackets, right? Not normal. They have some very specific distinguishing features that distinguish them from normal bacteria. But then all those topics that we will discuss on this and the next lecture, and the next lecture will be about rickettsia and prions, so are also included into the questions for examination. So that's why you need to know at least, maybe not so much about these types of bacteria. So we start today with chlamydia, and as you see here, and you see here the formation of the chlamydia inside the cell, and I would like to underline inside the cell, and so-called mycoplasma that look very nice here, that are free living bacteria, my dears. There is nothing common in these bacteria. Chlamydia and Mycoplasma are totally different. We place them in one single lecture just because we don't have time to read lecture about each one, the whole lecture, but not according to the common characteristics. Chlamydia is obligate intracellular parasite. So this is the property that make them the intermediate organism in between bacteria and viruses, because chlamydia cannot develop themselves outside the host cell. They are obligate intracellular parasites. That you have to remember about them. So what you have to remember about mycoplasma, they are free living organisms. We may grow them on the solid medium or liquid medium, but they are smallest bacteria, and they are as small as the largest virus, for example, pox virus. But still it is free living organism. So my dears, we step by step discuss our organisms today, and we start with chlamydia. So you can see here the members of chlamydia family, and due to the colored false color scanning electron microphotograph of a cultured human cervix cancer cell infected by Chlamydia trachomatis. Here you can see the inclusion body containing hundreds of chlamydial particles. And that's here you can see the attachment of the elementary body of chlamydia to the human epithelium. So first, few words about the classification of chlamydia. Why we start with classification? Many times classification of chlamydia had been considered already, and maybe it will be reconsidered in future. So because they are comparatively new pathogens to compare, for example, with syphilis and so and the chlamydial infections for very long while were underdiagnosed because it is very difficult to see them because they are protected by intracellular location. We didn't know about them for a very long while. So that's why they are comparatively new bacteria. And in order chlamydialis, we have several families. My dears, you don't need even to pronounce this. They are shown just for your information. But what we need to pronounce and know is family Chlamydiaceae. And here in this family, several genera are discovered, but then in this family, we may have human pathogens that now arranged in two genera, Chlamydia and Chlamydophila, my dears. Still, there are a lot of speculations about Chlamydophila genus. So in some books, you still can see all these three, Chlamydia trachomatis, Chlamydia pneumoniae, and Chlamydia psittaci, in one single genus of Chlamydia. In some other sources, it is believed that this classification had been developed in 2009, and then in 2011 they again put Chlamydophila back to the Chlamydia genus. My dears, that's a bit messy. Then, without referring to the particular classification, you need to know that we have three basic human pathogens there. Chlamydia trachomatis, Chlamydophila or Chlamydia pneumoniae, and Chlamydophila or Chlamydia psittaci. So you can understand this name, psittaci, it means avian chlamydia. So what are the characteristics common to all chlamydia that may cause diseases in humans? Regarding the structure of the cell wall, they appear as gram-negative bacteria. Then the second very important property why we call them, this is explanation why we call them obligate intracellular parasites. They are considered to be as energy parasites because they lack mechanisms for energy production. So it is believed now that they may perform some pathways of fermentation, but this energy is definitely not cannot cover all their requirements in ATP. So they cannot synthesize enough ATP molecules. So that's why they are obligate intracellular parasites, because they depend on the host cell energy. That's why they may develop themselves in the host cell only. So all chlamydia are similar if we take a look at them under the microscope. Their morphological features are indistinguishable within the order. And also they share the common group antigens. They usually are spherical or ovoid, as you see here, right? And regarding the structure of the cell wall, they have the peptidoglycan, but it is a bit not typical for bacteria because it lacks the unique component of the peptidoglycan of bacteria, that is N-acetylmuramic acid. So, still, we know that this atypical peptidoglycan is tetrapeptide linked with the polypeptides, so we call it tetrapeptide linked matrix. So there's not a conventional peptidoglycan. So, and as I said, out of the chlamydia that infect humans are trachomatis, pneumonia, and psittaci. What are some more common characteristics? The thing is that chlamydia in the body may occur in two distinct morphological and antigenic forms. So, and I would like to underline here that it resembles the virus, right? Do you remember that the viruses have two stages in their life cycle? Extracellular stage, when we call it a virion, and intracellular stage, when it get rid of its capsid and envelope and exists in the form of nucleic acid, viral genome, really, maybe with some very important proteins or enzymes that help in the transcription, translation, and all other things that happens in the cell. So chlamydia demonstrates something like this. Of course, not exactly, but that is the hallmark of chlamydia exactly. So they occur in the body within two forms. So first, they may exist in a shape of small infectious elementary bodies. You can see it here. They are infectious. It means they may bind with another cell and cause infection of the non-infected cell. So the elementary body are not spores, my dears, but like a spore, the elementary body is resistant to many harsh environmental factors. But the elementary body itself doesn't have that important metabolic activity. It is not replicable form of chlamydia. It cannot divide. But I would like to underline that it is infectious because it has all means, adhesins and everything to adhere to non-infected cell and infect host cell. The second type of form, or the second form in which chlamydia may exist in the human body or any host, that are the large non-infectious particles that are called reticulate bodies. See, they are here. And they are protected by intracellular location. They occur inside the cell only and protected by intracellular location. If they go outside the cell, they die. Now we discuss this very important characteristic of the chlamydia life cycle. So we call it special development cycle. My dears, you should remember always that this cycle is attributed to the multiplication of chlamydia. As soon as you remember this word, development cycle of chlamydia, you will be able to answer correctly on the examination. So in this development cycle, we can see that the infectious small elementary body adheres to the cell and enter the cell. Within first 2 hours, in the endosome, we have development or differentiation of elementary body into reticular body. That's like in spores. One spore develops vegetative cell. That's exactly like that. Elementary body, one gives rise to reticulate body. Then after 12 hours, we have within the next 10 hours, we have multiplication of reticulate body by binary fission. And the growth of inclusion inside the host cell. Then within next several hours, we may have the formation of the persistent forms of chlamydia. What does it mean, persistent form? These are the dormant stage of chlamydia that may persist in itself for a very long while, especially in harsh environmental conditions. But then one portion of the reticulate bodies associate with the inclusion membrane, and then the transformation starts when reticulate body will be differentiated into the elementary bodies. Then more elementary bodies will be appeared within 48 hours. That's how the whole cycle takes. And then we have the inclusion exocytosis when the endosome will be exocytosed and release the content into the environment. So altogether, it will be both elementary and inclusion, the reticulate bodies. Reticulate bodies die, so they cannot stand that extracellular location. And in elementary body are infectious, so they go and infect some other cells. This is the cycle. In the previous slide, you've seen a picture, simplified scheme of the process. And here you can see the electron microphotograph of Chlamydia trachomatis bodies in the cytoplasm of the host cell. Reticulate body, that's what we see here. There's a metabolically active, large, but very sensitive to the environmental conditions form. Elementary body is more dense, infectious form that doesn't replicate. And by the way, we also may see here the intermediate stage between reticulate and elementary body that is called intermediate body, where you can see the transformation starts already from reticulate to elementary bodies. And you can see more elementary bodies here formed out of reticulate body. So now just some description of the two types of the bodies. Elementary body is the environmentally stable and it is infectious particle. It contains membrane and the electron-dense nucleoid. And we refer to the structure of bacterial cell. They don't have nucleus, as you know, as you remember, might remember. They have nucleoid, this electron-dense part of the cytoplasm where chromosome occurs. Elementary body is small, 0.4 micrometers in diameter. And they have membrane proteins that are very important in adhesion. And these proteins are cross-linked by disulfide bond that can be broken, and the adhesins will be activated. Elementary bodies have very high activity to the host epithelial cells, and they have on their surfaces a lot of various adhesins like heparin sulfide-like proteoglycans, and the most important family is the family of major outer membrane proteins. And one variant of these outer membrane proteins are glycosylated outer membrane proteins, and some other individual polypeptide factors. So the mechanism of entry of the elementary body into the epithelial cell is binding of the elementary body through the surface adhesins, then receptor-mediated endocytosis, usually into clathrin-coated pits, and then pinocytosis, or another way is pinocytosis via non-coated pits. And the thing is that chlamydia inhibit the fusion of the endosome with the lysosome. So, and the outer membrane proteins prevent this fusion. So that's why lysosomal enzymes will be not able to digest elementary body. Then the reticulate body, this reorganization is performed from elementary body into reticulate body, and within the membrane-bound vacuole, the disulfide bonds of EB will be destroyed, cross-linkage will be broken, and the elementary body is reorganized into a large reticulate body. So you see here the development growth cycle within that is shown within the epithelial cell. So that's how it happens. So 6-12 hours formation of reticulate body, accumulation of the reticulate bodies. Then the rupture of the epithelial cell, by lysis of the elementary body, or exocytosis. So these two types of the release is possible. Reticulate body, what is it? You can see here reticulate bodies. So, and here's the reticulate bodies. So these are large cells, about up to 1 micrometer in diameter, and they devoid of the electron-dense nucleoid. So they grow in size first and divide by binary fission, just like bacteria. They'd also need to grow in size in twice before it divides by binary fission. Subsequently, elementary bodies will be derived or developed from the reticulate bodies to form a cytoplasmic inclusions. And you can see here unusual inclusions inside the cell, inside the cytoplasm of the cell. And the newly formed elementary body will be liberated from the host cell and can infect a new cell. That's the development in another way. So altogether, we may see several stages of this process. So, what do we know about the chemical composition of chlamydia? Because it is of gram-negative bacteria cell wall type, so we supposed to distinguish in the cell wall of chlamydia, the inner membrane and the outer membrane. But generally, the cell wall of chlamydia is quite rigid, but what is unusual, it has very high content of lipids. And as we said, it contains atypical peptidoglycan and quite a thin one without N-acetylmuramic acid. And the peptidoglycan matrix is cross-linked with the tetrapeptides. The thing is that in the transpeptidases that are the targets for penicillin, they are also found because transpeptidation is performed in the matrix. Of course, the targets for penicillin are there, but the effectiveness of penicillin in chlamydia is really very low. The cell wall formation can be inhibited by drugs, but it is not really effective. So then, another very important antibacterial factor of our body, that occurs in our body is lysozyme. But because there are no the link between N-acetylmuramic acid and N-acetylglucosamine, the lysozyme has no effect on the cell wall of chlamydia. So we suppose not to account on the activity and the lysosomal activity of macrophages. So inside the chlamydial cell, we have a circular genome, and it is similar to genome of other bacteria, but it is very small with molecular weight, 7 multiplied by 10 powered 8 daltons. How can we see chlamydia? So, what kind of tinctorial or staining properties they will develop? So, usually we may use Giemsa staining. After the Giemsa staining, elementary body stains purple and reticulate body appears blue. So, and then they form intracellular inclusions that are seen in the Giemsa staining. So they appear as compact masses near the nucleus of the cell, stained dark purple. And inclusion of Chlamydia psittaci are usually diffused intracytoplasmic aggregates that are different from those of Chlamydia trachomatis. So gram stain of chlamydia is not really useful in identification. Usually, rather, we use immunofluorescent tests. So, because these special antibodies can be produced against elementary bodies and reticulate bodies, and inclusions stained with group-specific, species-specific, and serovar-specific antibodies. I would like to underline these, my dears. So we may have antigens in chlamydia, typical for large groups of chlamydia, then typical for species, and typical to some groups within species. So we come up to when we come up to Chlamydia trachomatis, you can see the antigenic diversity of Chlamydia trachomatis. So we also may perform iodine staining, and that's typical for Chlamydia trachomatis to develop so intracellular inclusions will turn brown only because they accumulate glycogen matrix and that's why they are stained brown by Lugol staining. So that's now a few words about the antigen that we mentioned already. So the antigens of chlamydia can be detected by immunofluorescent test. That's the best one. That's how we see them under the fluorescent microscope, where the inclusions are stained orange, maybe by acridine orange or some others. So the given organism may contain several specificity antigens. So when we are talking about genus-specific or so-called group-specific antigens, this is usually liposaccharide antigens. Usually they can be detected by immunofluorescent test or complement fixation test, my dears. CFT, this is the complement fixation test. The antigens of narrow specificity. These are species-specific, common in species, and serovar specific antigens. And they are mainly outer membrane proteins. That means with the species-specific antibodies, we may distinguish three species: Trachomatis, Psittacia, and pneumonia. And those species-specific antibodies will reveal species-specific antigens. But then within the species, based on the antigenic structure of outer membrane proteins, we may distinguish some serovars. That will be serovar-specific antigens, usually also protein antigens. So within Chlamydia trachomatis species, we distinguish 18 serovars. Chlamydia psittacia has from 8 to 13 serovars, so different antigens, serovar-specific antigens. In class 3, Chlamydia pneumonia has 4 serovars of 12 biovar, causing infections in humans. My dears, we won't touch so much the class 3 Chlamydia psittacia pneumonia, but it is really very important for Chlamydia trachomatis. Why you medical doctors should bother whether it has 18 serovars or more? The thing is that Chlamydia trachomatis causes usually serovar-specific diseases. What does it mean? It means that the particular clinical symptoms or particular nosological form of disease is associated with particular serovars. Then a bit later on we will see the demonstration of it for Chlamydia trachomatis. So then that's how we may see the elementary bodies that are located outside the host cell, right? EN inclusions labeled with the monoclonal antibodies inside the human cells. Few words about the detection of nucleic acid. So the method is applicable, that is nucleic acid probe method or hybridization. For example, Chlamydia trachomatis have 10 powered 4 copies of the 16S rRNA per particle, and these are species-specific sequences. They can be detected using a chemiluminescent DNA probes that hybridize to the 16S ribosomes. The also polymerase chain reaction is used when we perform nucleic acid amplification. And also ligase chain reaction is possible in identification of Chlamydia trachomatis. So it is believed that these tests, first, they are more rapid, and of course, they are more sensitive than culture and not amplification, non-amplification tests. Why it is so important to understand? Because chlamydia causes so-called, as we said this word before, persistency. It means it can persist inside the human cells without coming out of the cell, so it cannot be identified by any other methods, and without showing symptoms of the disease. So in a sort of dormant or resting state. And that's why it is really very difficult to identify chlamydia within the stage of they usually cause chronic infections, and within the stage of remission, it is very difficult to understand whether they are still in the body or not. How can we grow them? So because they are obligate intracellular parasites, the growth should occur within the host organism inside the cells. So for this reason, we may use two types of growth, at least. All chlamydia grow very well in embryonated hens' eggs, and usually we use yolk sac for their growth. And usually it yields a large number of chlamydia. So we also may use the cultures of eukaryotic cell lines. Mainly we use for this the cervical cancer cells that are HeLa. Also we may use McCoy cells, treated usually with cycloheximide commonly. And for chlamydia pneumonia, we use HAP cells or HL cells. So, what antibacterial drugs may be effective against chlamydia? Generally, we may use many drugs, but first, they're supposed to be of broad spectrum. Do you remember when we studied antibiotics, we underlined that we may have antibiotics with narrow spectrum and broad spectrum. Usually the broad spectrum antibiotics are used for chlamydia because nobody developed really the separate group of drugs that may affect chlamydia only but not the others. So the drugs are effective that inhibit the protein synthesis, tetracyclines and macrolides, and they are quite effective in clinical infections. So, out of those drugs, the azithromycin is usually very common. So then sulfonamides, sulfadimidine, inhibit Chlamydia trachomatis strains because these organisms synthesize folates as regular bacteria, not like human cells. But cell wall inhibitors are not really effective. They develop, I mean, there is a target for penicillins and cephalosporins in the cell wall of chlamydia. But in clinical disease, penicillin is not effective. And even very, so make the disease worse because it may cause the development of the morphologically defective forms that can be difficult to diagnose, but then that can still cause persistent infections. So the good group of aminoglycosides are not also effective against them. So this is explanation of why until now, from now, we consider chlamydial infections are very common. And by the way, the truth is that start to be much more common than traditional sexually transmitted diseases, for example, like syphilis or gonorrhea. And chlamydia is one of the infections that are rising, the incidence of which are rising in the population, in the world population. The problem is in persistence of chlamydia in tissues. And the thing is, when they persist, they can cause just subclinical infections. So without any pronounced symptoms like we have, for example, in gonococcal infections. So, and because they are protected by intracellular location, they may persist inside the cell in the presence of high level of antibodies, high titers of specific antibodies. It means antibodies will be developed, but they cannot protect organism against infection, and they cannot really help to eliminate chlamydia from the body. Overt diseases is not common in the natural hosts of the organism. So spread from one host species to another may lead to the clinical manifestation of the disease. For example, psittacosis is a very good example when the disease is transmitted from parrots or some other birds to humans. And when the treatment of chlamydial infections can be successful. And that is a good idea, right? Early and intensive treatment with antimicrobial drugs for prolonged period of time may eliminate the organism from the host. And late treatment with antimicrobial drug in moderate doses may suppress disease but permit persistence of chlamydia. But then, my dears, to start early and intensive treatment, we have to improve and develop the correct diagnostic procedure. But if usually within the persistency there are no clear clinical symptoms that may make us thinking of chlamydiasis, so the symptoms quite often appear when the persistency is established in the cells of the body. So I mean that there's no chance to get rid of the chlamydia. So that makes chlamydial infections right now very common and really dangerous regarding the development sterility or infertility in patients. So now we take a look, and I would like to show this first. So that's something about serovar specific diseases. You can see here that Chlamydia trachomatis is divided into two types of infections: invasive uh, chlamydiasis and ocular genital. And ocular genital is divided into genital diseases and ocular diseases. So, and all these three groups of diseases are produced by particular serovars. So that's what we mean under serovar specific diseases. Chlamydia trachomatis of serovars L, several types develop invasive lymphogranulomatosis. Then serovars from D to K, I and J, they cause genital infections. And serovars may be A, B with variants, and C cause ocular diseases. So now we go for chlamydia trachomatis And now we are talking about serovars that cause ocular infections. So the natural hosts for it are humans. And disease in humans is trachoma, or in another word, Chlamydia keratoconjunctivitis. So it also may cause inclusion conjunctivitis of newborns, and some serovars may cause sexually transmitted diseases, non-gonococcal urethritis, symphysitis, cervicitis, then infant pneumonia, and also lymphogranuloma venereum. And this out of 18 serovars, we have the spectrum of the disease. So, usually it may be inhibited by sulfonamides. And the transmission may occur of Chlamydia trachomatis from person to person, mother to infant. Then inclusions usually compact, round, vacuolar, glycogen occurs, and we reveal it by lubule staining. That's common for all these species specific properties. So few words about the virulence factors of Chlamydia trachomatis. Except for adhesin that we mentioned already, that are connected with heparin sulfate-like proteoglycans and major outer membrane proteins, glycosylated and not. So they may cause stimulation of receptor-mediated endocytosis and phagocytosis of macrophages. So they also inhibit the lysosomal fusion. So then, because it is gram-negative bacteria, it demonstrates lipopolysaccharide endotoxin, and lipid A will be still there. And also they may produce some exotoxin that are thermolabile, so they are proteins. And they are toxic, for example, for mice. That's our table for several specific diseases. So we start with the diseases. And first one is, of course, trachoma, the ocular disease. It is endemic chronic keratoconjunctivitis caused by specific serovars that we mentioned here, right? So, and throughout the world, around 80 million people have an active infection. In some endemic areas, infection may be present in as many as up to 90% of children, because usually children are infected in the very first years of their life. Trachoma, and it may cause also blindness. Trachoma is spread by direct or indirect contact with eye, nose, throat secretion. From infected individuals, and very common, it may be carried by flies. The disease starts with the acute inflammation in the conjunctiva and cornea and progress then to scarring and blindness. Initial infection occurs, as we said, in the very early childhood, and usually the disease is not that dangerous by itself, but the long-term consequences can be as a result of this infection. That is mainly blindness. And now you can see the map with the newest data that are available right now is from 2018, my dears. As I think, generally, as I said once already, that is very difficult to get fresh global results about global epidemic of the disease, because it is connected with the difficulties in collecting data from all around throughout the world from different countries. But then I think within our days it is going even worse because of the coronavirus infection within last 2 years. It is very difficult to collect the fresh epidemiological information in other diseases. But what I've got already for 2019, that in 2019, so the reduction on over the trachoma infections is shown by WHO. You can see here that in 2002 the number of people at risk fell from was 1.5 billion, and in 2019, so it drops to 142 million, so 91% reduction. And in 2019, it was shown that in 8 countries, so, and they are validated by WHO for achieving elimination of trachoma from the human population. That is very good for those countries where it is endemic, and you can see Africa, Asia, Central and South America, and Mediterranean basin. So, clinical findings of trachoma. Usually, the disease will be developed 3 to 10 days since infection. That is incubation period. So, the earliest symptoms are lacrimation, mucopurulent discharge from the eye, conjunctival hyperemia, and that is very typical follicle hypertrophy that you see here, and keratitis, subepithelial infiltration, and so-called panus formation, that is extension of limbal vessels into the cornea. The panus extension causes scarring of the conjunctiva and eyelid deformation. You can see it here. And bacterial superinfection causes blindness of the eye usually. So usually this is a local disease. We don't see any systemic uh, infection, the signs of any systemic infection. But that is very serious consequences of the disease. Usually we use sulfonamides for treatment or macrolides that are very effective. Periodic topical application of the drugs to conjunctiva of all members of the community is supplemented with oral administration. Treatment options include oral azithromycin and topical tetracycline. So the thing is that chlamydia can persist in the body during and after drug treatment, and recurrence of activity of chlamydia is common due to the persistency, as we said, so it's very difficult to get rid of it. So then, few words about other variants of chlamydia trachomatis infections. These, as we stated, disease caused by L serovars. So here they are numbered L1, 2, 3, and in our table that are labeled with small Latin letters. So can you see there? Oh, here, see? A, B, and and C, but it doesn't matter so much. So this is a sexually transmitted disease caused by particular L serovars or Chlamydia trachomatis. The disease is characterized by separative inguinal lymph nodes at this initial stage of it, and then bulbous inguinal adenitis, bulbous abscesses formation, and proctitis. So these is the very severe disease that initially can be misdiagnosed with Yersinia pestis infection because of the similarity of the bulbous formation. The serovirus DGHJ are spread everywhere, and maybe they make the commonest genital infections. Transmission is from person to person through sexual contact, but also it can be transmitted from mother to child. So this disease is called genital chlamydiosis. That's how they are called, referring to the common sexually transmitted diseases. So usually they cause non-gonococcal urethritis, salpingitis, and cervicitis. Inclusion conjunctivitis of the newborns when transmitted to fetus, infant pneumonia, and otitis also are caused by those serovars. How can we perform the diagnosis? So for serology, indirect or immunofluorescent assay, hemagglutination assay, ELISA now is maybe the commonest test for these types of chlamydia. Diagnosis may be confirmed in adults by antibody titers, and in newborns by anti-chlamydial IgM because they cannot be transported through the placenta, and that's not maternal but baby's antibody. So the prevalence of the chlamydiosis, and these are diseases caused by serovirus D, G, H, and J, exactly. So, and that's usually mainly the sexually transmitted disease. Usually is estimated together with the other diseases like most common sexually transmitted diseases in the global statistic, together with gonorrhea, syphilis, and trichomoniasis. Then, if you take a look at the chlamydial infections that are given here in percent and the variations in male to female, you can still see here that the percent of infected individuals is very high. Especially it is very high in the Western Pacific region and the region of Americas, up to 17.6%, and this is in the adult population from 15 to 49 age groups. Okay, from 15 to 89. So if you take into the European region, it is a bit lower, and Southeast Pacific Asia region and in Eastern Mediterranean region. But it is quite high in Africa region, Western Pacific region, and Americas region. So now a few words about chlamydia pneumonia, or in nowadays variant of classification, chlamydophila pneumonia. Usually natural hosts are humans, and in humans the special biovar occurs. Biovar TVAR, Taiwan Acute Respiratory Biovar, because it was initially identified there. So the disease caused in humans are respiratory tract infections, pneumonia, bronchitis, pharyngitis, sinusitis. And animals also may develop some diseases over the animal's biovars. So they resist sulfonamides. That's why macrolides or tetracyclines usually are used for treatment. Transmission occurs through as airborne disease transmitted from person to person. If we take a look at the inclusions, they are different from that caused by Chlamydia trachomatis. They are round, dense, and glycogen is absent. These are the inclusions. So, in Tvar Biovar, we have 4 serovars. And according to the serological surveillance, 60 to 80% of adults are seropositive with the Chlamydia pneumonia. And it is believed that this type of infection correlates with the coronary atherosclerosis. So they affect cells of respiratory epithelium and also endothelial cells. For the diagnosis, usually a Lee-Sand polymerase chain reaction is used. So, and finally, we have Chlamydophila psittaci. And that's we see the typical way of the infection. So, the transmission usually occurs as airborne disease with bird excreta from birds to humans. Initially, it had been called psittaci because the parrots were recognized as the commonest source of this infection. But then it appeared that birds of many species can harbor Chlamydophila psittaci and also can be uh, recognized as a source of infection if they are kept mainly at home and have the uh, as pets, right? And the people communicate tightly with these birds. So, if we are talking about the type of disease caused in humans, so this is pneumonia, or it is recognized as fever of unexplained origin, because usually the diagnosis ornithosis or psittacosis is very difficult to develop, uh, and maybe some epidemiological information may have start thinking of psittacosis. Usually, this type of disease is very much underdiagnosed. So, in natural hosts, they may cause ornithosis in birds. They may also cause some infection in uh, cats and some other diseases in animals. This type is resistant to sulfonamide, so other drugs supposed to be used for treatment. The inclusions produced by Chlamydophila psittaci are usually variable in shape, so they are pleomorphic, so they are dense, usually small, and glycogen is again absent in this inclusion. So if we stain it by Lugol staining, we won't get the appearance of these inclusions. We may develop at least from 8 to 13 serovars of this chlamydia. So it can be recognized because it is quite rare disease. So they may be recognized by finding of antibodies or antibody titers, and the very low titers, 1 is to 16, will be uh, recognized as positive, because I would like to repeat, there's a quite rare infection and the cut level of antibodies is very low. Then the regular diagnosis is done by immunofluorescent, ELISA, and PCR test. But also, due to the hypersensitivity reaction, skin test with ornithine also may help to diagnose the hypersensitivity reaction to the antigens on Chlamydia psittaci. With this, I would like to finish with chlamydia, and I would like to repeat that chlamydia are obligate intracellular parasites because they lack ability to develop energy or ATP molecules enough for metabolic activity by their own. So that's why they depend on the host cell machinery to develop ATPs. So, and again, the most important things to remember about chlamydia are their development cycle, that is differentiation from infectious elementary body to reticulate body. And the second important thing to recognize, to remember, is the species specific serovirus specific clinical manifestation of the diseases, the table that we see here. With this, I would like to finish with chlamydia and go for mycoplasma. And we know that mycoplasma are smallest bacteria, and they are recognized as smallest self-replicating organism. They can be free living in nature. So they are of the size of them is 0.250 micrometers in diameter. My dears, this is the size of one of the largest virus. So, and it consists of the membrane that is enriched in lipids. And then it is important that within the cytoplasm we may find the so-called soluble RNA molecules and also DNA chromosome within the part of this cytosol, so this nucleoid. Then plenty of soluble proteins inside the cytoplasm and also ribosomes that are important in the protein generation. So the general structure is very simple. But then still I would like to underline that it is free living organism. What are the common characteristics of mycoplasma? So as we said, smallest self-replicating organism. It means they don't need host cells to multiply. They may produce everything they need by their own. It is believed that mycoplasma have reduced genome, and the smallest genome of certain mycoplasma is little more than twice the genome size of the larger viruses. So that's very little genome, very small genome. One more very important feature of mycoplasma, that they are cell wall free bacteria, my dears. They don't have peptidoglycan. They don't have cell wall. That's why if you take a look at the electron microphotograph of mycoplasma, you can see that they are highly pleomorphic, so they don't have rigid cell wall. That's why they are highly pleomorphic. But instead, they have a triple layered membrane enriched in sterols or generally lipids. So they need lipids for their life, higher content than the other bacteria. So, and that's why if we are going to grow this organism, we have to add sterols into the culture medium. And because they are cell wall free organisms, they are resistant to penicillin. because there is no target. Transpeptidase is a target for penicillin cephalosporin. So there is no target for penicillin drugs. So that's why we consider that mycoplasma are naturally resistant to penicillin drugs. Go ahead with some more common characteristics of mycoplasma. So they can be inhibited by broadest spectrum antibiotics, tetracyclines or macrolides. And I would like to underline that they can reproduce in cell-free media. And here you can see the colonies that are called fried egg colonies. Then we take a look at them one more time. So, because the appearance of the colony looks like the fried eggs. So, you can see that center of the colony is embedded usually beneath the surface. And the thing is that growth of the organisms is inhibited by specific antibodies because they don't develop, they don't demonstrate the intracellular properties. I mean, they are not intracellular, obligate intracellular parasites. So because they need sterol, they demonstrate very high affinity for mammalian cell membranes enriched in phospholipids. So that's why mycoplasma can be called membrane parasites. They cannot be studied by usual bacteriological methods. Why? Because of their very small size, and that's why the colonies are very small also. Then the plasticity of the cells and the pleomorphic properties of the cell also make it very difficult. And because they don't have cell wall, they demonstrate poor staining with aniline dyes. So that makes mycoplasma very poor model for culture method. There's so many mycoplasma, but which of them are of medical importance? So for the first we have to mention mycoplasma pneumonia, and due to the name you understand that they cause usually pneumonia, but as a result of uh, uh, cross reactivity, they may cause some myocarditis, joint infections, and other infections complicated by hypersensitivity reactions. Mycoplasma hominis is associated with postpartum fever and uterine tube infection, that is salpingitis. Ureaplasma urealyticum causes non-gonococcal urethritis in men and lung diseases in premature infants of low birth weight. So Mycoplasma genitalium is closely related to Mycoplasma pneumonia. It's difficult to distinguish them and has been associated with urethral and other infections. And this is a classical shape of pathogenic Mycoplasma. This is a flask shape, right, with a narrow neck. And again, this is distribution of the Mycoplasma pneumonia that causes pneumonia, joint infection, myocarditis, and other diseases. And if we roughly take a look at it, we can see that here we have very high, so this is the 22 to 84 for 100,000 population. But then I think that in many countries, this disease is underdiagnosed yet. So because this, again, this is very difficult to diagnose this disease. Few words about the morphological and tinctorial properties of mycoplasma. We may perform dark field examination, so that is an example of dark field examination. And we also may use gene staining and immunofluorescence to stain. Morphology appears different according to the method of examination. For example, depending on where bacteria are taken from, solid media or liquid media, it may develop different morphological features. Growth in fluid media usually gives rise to many different forms. You can see rings, bacillary, and spiral bodies, filamentous bodies, and granules. But growth on solid medium consists of protoplasmic masses of indefinite shape. They range from 50 to 300 nanometers in diameter. So we can see here, that's like what you may see there. See? Then, if you take a look at mycoplasma infected cell, you can see here that the cell is covered. So all these small bubbles, these are mycoplasma cells that adheres to the membranes with their place where all adhesins are collected in flask-like structure. And usually they are of irregular shape. So how do we grow? If you would like to grow mycoplasma, how can we do this? Usually we cultivate in heart infusion peptone broth with 2% agar and 30% of human acidic fluid, or the animal acidic fluid can be used, rabbit serum, horse serum, or acidic fluid. They grow not very fast, so we need to keep them in the incubator up to 96 hours to get some growth. Genes stain of the centrifuge sediment show the characteristic pleomorphic structures. So subculture on solid media yields the very small minute colonies after 2-6 days on biphasic broth of agar and agar media. Colonies are measured up to 50 micrometers in diameter. They are very small, different to detect. So they are round with a granular surface and dark center buried in agar. So I have one more picture of it. See, that is very nice mycoplasma colonies on this special medium with fried egg appearance. See, looks very nice. And also, that's not so. Growth characteristics. Because mycoplasma are unusual, so their size is considered to be extremely small, and they are compatible to chlamydia or large viruses. So they grow on cell-free media. It means cells, we don't need cells for their growth, living cells for their growth. But we need to add sterol because they have extra sterol requirements. So they are resistant to thallium acetate, so it can be used as a selective component in the medium because it inhibits the growth of other bacteria. They can use the glucose as a source of infection. Ureaplasma usually require urea in the medium. Mycoplasma may produce peroxidase and hemolyze red blood cells, my dear. So small, but it may produce hemolysis. So the antigenic structure of mycoplasma is very complicated because the species of mycoplasma are antigenically distinct, and some species can be divided into serotypes. So usually in different diagnostic tests, the different types of antigens are used. For ELISA, the antigens are glycolipids and proteins, and for complement fixation test, glycolipids. So the species, the clue properties that are used for classification, for identification of species are biochemical and serological characteristics. At least 15 species can be identified in humans. You can see their names, but most important, medically important, are Mycoplasma hominis, pneumonia, genitalium, and Ureaplasma urealyticum. Pathogenesis of Mycoplasma infection. First, they adhere to the host cell by adhesins, that is, interactive proline-rich protein that are located mainly on the polar tip structure of the flask-like mycoplasma. And mycoplasma attached to the surface of the cells through the mucosal cell sialoglycoconjugates and sulfated glycolipids. And then, as a result, they may stay at the surface of the cells, and there they may produce direct cytotoxic activity. It means those very small bacteria may cause the cell death directly. Why? Because it is believed they produce cytotoxicity through generation of the peroxide, hydrogen peroxide and superoxide radicals that perforate the cell membrane of host cell and cause the osmotic lysis of the cell. And then they may perform cytolysis mediated by antigen-antibody reactions. See if the antigens are found of mycoplasma or mycoplasma themselves cover the host cell, so the antibodies can bind with it and the lytic mechanisms will be activated to destroy this cell. And also mycoplasma compete for the sterols with the cell, compete and cause depletion of the sterols in human cell. Here you can see the more broad spectrum of the virulence factors or what kind of damage mycoplasma may cause in human cells or in mammalian cells. See, this is the what factors of mycoplasma pneumonia are important in pathogenesis of this disease. So, direct damage mechanism of the cell by adhesion damage, membrane fusion damage, nutrition depletion of the sterols, invasive damage, and toxic damage. And this is direct damage produced by mycoplasma. But then we may have indirect damage or immune-mediated damage of the cells that are covered with mycoplasma. Humoral immune mechanisms that the variant with antibody that we discussed already. Cell immune damage mechanism, because the infected cell can be the target for T cytotoxic lymphocytes. Then inflammatory damage, because it releases superoxide radicals, and those radicals also cause permanent inflammation in the body. Then antigen immune damage and immunosuppressive damage, because it may cause hypersensitivity reaction. These are the factors of the cell damage attributed to the pathogenesis of Mycoplasma pneumonia diseases. So you can see here the typical shape of pathogenic Mycoplasma pneumonia, where they may produce flask-like structures here and here. There's a very nice picture, right? And the filamental shape also. And on this tip of the flask-shaped structure, we have all adhesins and accessory proteins. So diagnostic laboratory tests. Rapid diagnosis can be produced by polymerase chain reaction. Microscopic examination is useless for specimen taken from the patient because they are very small and cannot be seen really. Usually, the culture method can be used. We culture them on special solid media for several days in microaerophilic conditions. Then we may get the picture like this with fried egg appearance of the colonies. And the material may be inoculated in the special broth and incubated aerobically. So usually several sub or passages are needed to accumulate bacteria for microscopic examination. So epifluorescence is used as detection of Mycoplasma infection within the cell line. Infected cells cultures are co-cultured with an indicator cell line, that is a virus cell, for 2 or 3 days before staining with fluorochrome dye, which binds specifically to DNA. Stained infected cells may be viewed under immune epifluorescence. So then serology can be used, antibodies can be demonstrated, so by complement fixation test or indirect hemagglutination test. So the test for growth inhibition by antibodies. These are counter immunoelectrophoresis that can be used. And I would like to show you the new one. So, and this is Mycoplasma Diagnostic Laboratory Test. So that is the rapid test that can be used for detection of Mycoplasma. But the advantage of this test is that it is very simple and complete diagnosis of urogenital Mycoplasma, but in the same time, not only Mycoplasma can be identified, but also the identification of antibiotic susceptibility testing for detection of Mycoplasma hominis and ureaplasma can be performed within this single test. So, what issues of treatment and prevention do we consider? The treatment can be performed by a variety of antimicrobial drugs. They are all quite effective against Mycoplasma, but except for the beta-lactam drugs, because there is no target in Mycoplasma for beta-lactam drugs, it develops natural resistance to them. So usually tetracyclines and erythromycins and macrolides are drugs of choice in Mycoplasma pneumonia. So it is believed that some Mycoplasma, especially ureaplasma, develop resistance to tetracycline already. Most strains of Mycoplasma are resistant to many beta-lactam drugs. But what is common for both chlamydial infection and Mycoplasma infection? By now, we don't have any means for specific prevention or prophylaxis. By the way, I read about a lot of vaccine candidates, and the laboratories are working on that. But due to the antigenic variability of these microorganisms and due to the intracellular location of mycoplasma, of chlamydia species, it is still no preparations that demonstrate the protectiveness for diseases is not available. So these are infections that cannot be non-preventable so-called infections. So the only way to prevent this infection is if you are talking about sexually transmitted diseases, so using barrier techniques in the as a means of general prophylaxis. With this, guys, I would like to finish my lecture. We work without the break, so that's why we have discussed all that we wanted. And with this, I consider uh, the lecture is finished and you guys free as birds. I would like to repeat that the topic of today's lecture, the questions, chlamydia and mycoplasma, are included into the exam questions. This is a part of our examination. So please read and be prepared. There will be no classes in chlamydia and mycoplasma. There is time for lectures in these subjects, but no time in classes. So that's why you read what you have in the lecture book, lecture, and medical microbiology books. With this, I would like to say goodbye to everyone. See you uh, next Monday with a lecture about rickettsia and prions that are somewhere. We don't even know where they are, what kind of organisms are they. We don't even call them organisms. So please, waiting for you next time. Goodbye, guys. Have a nice day.